Well, okay, so it's almost 5 minutes past 2, so it's time to start our Sano seminar on computational medicine. And this seminar is the third one from the series of, let's say, seminars on Nobel Prizes. And today we have a guest from such an institution like Concept Life Science, a research institution in UK. And this is Dr. Zandile Nare. We were looking for a good speaker for this topic, for a topic on Nobel Prize in Physiology or Medicine in 2024. And one of the person who put a lot of effort in finding such a prominent speaker is a researcher from Sano, Paulina Orek. So I think that it would be good if Paulina will introduce Zan to participants of the seminar. Paulina,
Hello,
floor is yours.
Zan. I'm very glad that you will share your knowledge with us today. And Zan is a senior scientist at Concept Life Sciences. Concept Life Sciences is a company which plays the role of a partner for drug discovery challenges. They have like a huge experience and scientific knowledge in the areas of small molecules, cell and gene therapies, and biology. And the organization of the seminar in collaboration with Concept Life Sciences company was possible thanks to Dr. Karolina Tokarczuk, who is here. And Carolina, I met Carolina during my PhD studies. We were working together at the laboratory at the same team. And I got the information from Carolina that she has a colleague who has a great, huge knowledge in the area of the Nobel Prize of Medicine. And in the same time, Zan is like amazing speaker. So we decided to invite her to give the talk. So Zan, we would like to hear the presentation from you.
Thank you.
Thank you very much, Paulina. Zan, screen is yours. And if somebody would like to ask question during the presentation, please put it on questions and answers and also on chat, and I will try to such an immediate question transfer to Zan. And of course, there will be a discussion after Zan talk. So Zan, once again, screen is yours.
Thank you. Thank you, Marion and Paulina, for the introduction. It's very kind words from Carolina and yourself. Carolina is actually sitting right there behind me. You can't see her because she's blurred out. But yes, today I'm going to be talking about the Nobel Prize in Physiology or Medicine. So this was awarded to Victor Ambrose and Gary Rufkin. So the working title of the presentation is Tiny Titans, How MicroRNAs Are Reshaping Medicine. But as a maybe a caveat, I am not a microRNA expert or a developmental biologist. I'm coming from this as someone who is interested or like really interested in microRNAs and their application in medicine, and more specifically how we can use biophysical tools to learn more about these fascinating small molecules. So this is just the outline of my presentation today. So I'll give a brief background on the discovery of microRNAs. Then I'll talk about their implication in disease and how they can be exploited for future therapeutic developments. Then, as I said, I'll go on and talk about biophysical tools that we have here at Concept Life Sciences to assess microRNA-based therapeutics. And then I'll finish up and hopefully we'll have some time for questions and answers. As Marion said, just interrupt if you have any questions, and yeah, we can also just interrupt mid talk if that's easier. So yes, this work, as I said, the Nobel Prize was awarded to Victor Ambros and Gary Ruvkun for their work in discovering microRNAs in C. elegans. So this is a picture of what C. elegans look like. This is a nematode, a soil-dwelling nematode, and a powerful model organism that's been in use in science since the early 70s. So C. elegans was initially proposed as an ideal model for developmental biologies for different reasons, mainly because it has a highly predictable and consistent developmental pattern. This is what they call an invariant body plan. It's also transparent. If you look closely, the body of the worm is transparent, and so it's more easy to distinguish changes in different organs and sometimes different cell types. And the worm itself is relatively easy to genetically manipulate, so opening up opportunities for mutagenesis screening. Culture is cheap, so they're relatively cheap to maintain in the lab. They have relatively high sequence homology to the human genome, so meaning that discoveries that are made in C. elegans can be inferred to humans. They have a short lifespan as well, so this allows us to study multiple generations in a short amount of time and study evolution and aging. And also, this organism is amenable to high throughput sequencing and screening. C. elegans is now used in many different types of research, so including neuroscience, for example, to study neuronal circuits, genetics to study evolution, and in aging, as I said. So this discovery of microRNAs has really revolutionized biology, or our understanding of biology and disease, and I'll go on to discuss how and why that's happened. So below here, I have a timeline of the discovery of microRNAs, just to show that it's been, since the 80s, really since the 70s. But this timeline, which I got from BioRender, shows the timeline from the 1980s. And as I go through the talk, I'll talk through the different milestones. So starting, as I said, in the 70s, a mutagenesis screen in C. elegans led to the identification of a Lin4 gene mutant and the striking phenotype in this mutant. So you can see at the top here is the wild type nematode, which looks as you would expect. And then we have the Lin4 mutant, as I was saying. So you can see already that there's a difference, and the main differences were that many cell types and morphological structures were entirely absent, and also eggs accumulated in the worm because it wasn't able to progress to the next developmental stage. So the disruption of this development suggested that Lin4 likely encoded for a master regulator of developmental timing. And so in 1980, Ambrose discovered the Lin14 mutant, discovered that the Lin14 mutant has a developmental timing defects that were the opposite of what was seen in Lin4. So you can see the Lin14 mutant is small and it lacked larval development. And Ambrose later discovered that Lin4 is a negative regulator of Lin14. And I should say all these milestones I'm discussing, I'm not mentioning that some of it happened, in different lab groups. Some were discovered by Ambrose, some were by Ruvkun. If you want the detail of who's who, I would suggest you watch the Nobel Prize lecture that just happened, I think, a couple of weeks ago. I watched it and it was very interesting. But just to say that, if I miss someone's name out, it's not on purpose. So then after Ambrose found that out, Ruvkun identified Lin14 as a nuclear protein with stage-specific expression in development. So he found that expression of Lin14 protein was high in the L1 stage, and this was altered in Lin14 and Lin4 genetic mutants. So then they, at this point, worked in different labs. I think they had their own labs, so they set out to clone their respective genes by what they said was walking along the chromosome. So they were cloning shorter and shorter sections or fragments of the chromosome and analyzing the open reading frames. So the open reading frames are what is translated into a protein. So if an ORF can if you analyze it, you can sometimes see that it's not going to be translated because there's maybe a stop codon in the middle, or there's not a proper start codon, for example. So they analyzed these open reading frames to see if these fragments could rescue the observed phenotype. So I should also say that while they were doing this analysis here of walking the chromosome, when they were cloning shorter and shorter fragments and not seeing any effect that they expected, they then began to suspect that the genes that they were analyzing, Lin4 and Lin14, were short non-coding RNAs. And so when they came to probe for these transcripts by northern blotting, they identified in the wild type, you can see in this northern blot here, the wild type, you can see a band that corresponds to a 21 nucleotide sequence of Lin4, but this is absent in the Lin4 null mutant. However, when the nematodes are treated with Lin4, the Lin4 nucleotide microRNA, this rescues the phenotype. So we actually they saw a 60 nucleotide sequence, which is actually a precursor of the mature Lin4 microRNA. And so this confirmed that this short amino acid sequence rescued this phenotype. So next, after independently cloning their respective proteins, they compared sequences. So I don't know if I mentioned before, but they, I think, worked in the same postdoc lab, and then where they've both studied Lin4 and Lin14, and then they went off onto their respective, to form their respective research groups, but they still kind of maintained communication. And at this point, they shared their sequences and noticed that there were some suspicious regions of complementarity. So they later showed that the Lin4 microRNA formed some base pairing with elements of the Lin14 messenger RNA in the untranslated region. So this is the 3' UTR. And so this was a really groundbreaking discovery, and Ambrose later went on to show that this phenomenon extended to other nematode species. So this brought us up to the early 90s. So again, this is maybe a nice example of collaboration in science. Sharing is caring and productive. So then 7 years later, LET7 was identified. So this is also a short 21 nucleotide RNA that was complementary to the 3' UTRs of various genes. And the identification of this second microRNA suggested that these microRNAs may play a broader role in gene regulation. But unlike Lin4, LET7 was shown to be evolutionarily conserved across different species. So if you see on this phylogenetic tree that was built based on data that at that time, you can see here in the red, these are species that do not express microRNAs, but everything with a green plus does. And then everything with the blue dev suggests that these animals do not express microRNAs in the early life stages, but they do late in adulthood. So the fact that LET7 was evolutionarily conserved garnered a significant interest in microRNAs as post-transcriptional regulators of gene transcription, and this was in 2000. And so a vast number of different regulatory microRNAs have been identified now in animals. And as I said, these play an important role in regulation. And advances in molecular biology and sequencing technologies have now led to the identification of thousands of microRNAs in the human genome. And we also, from that, we have now this MIR base. This is a database for microRNA genes, and this includes over 48,000 mature microRNA sequences in 271 organisms. I'm going to skip over the discoveries in 2002, but just briefly to say that microRNAs in 2002 were then linked to cancer with identification of MIR15 and MIR16. And I should say that at this point, once the database was developed, the nomenclature to refer to different microRNAs was MIR and their associated number. So these two microRNAs were associated with lymphocytic leukemia in patients. And then dicer is one of the proteins involved in microRNA biogenesis. So dicer 1 syndrome was identified and linked to microRNA production as a tumor risk. And I'll go on later to talk about the future applications of microRNAs as biomarkers, as therapeutic agents, and potentially in agriculture. But just like to kind of talk about how this discovery has kind of revolutionized what we know about molecular biology, and referring to the central dogma of molecular biology. And this is a key rule that was, I think, first described by Francis Crick. And it was quite funny when I was preparing for this presentation. You know, biologists all know about the central dogma of molecular biology, but I hadn't actually read the paper. I just know that, you know, DNA is translated, or DNA is transcribed by RNA polymerase into messenger RNA, which is then translated by the ribosome into amino acids, and these assemble into proteins. And depending on the proteins, the location of the proteins in the cell, the location of the proteins in cells, this then decides cell and tissue fate. And in the paper, interestingly, Francis Crick starts off by stating or quoting Howard Temin, where he says that the central dogma that Francis Crick had previously described in 1958 as the keystone of molecular biology ever since, or has been the keystone of molecular biology ever since, is likely to prove a considerable oversimplification. So I read this and I was like, well, it's quite funny foreshadowing maybe for what was identified in this in Ambrose and Rufkin's work. So following on from what they discovered, and just to reiterate how microRNAs work, DNA is still transcribed into messenger RNA, but microRNAs bind complementary sequences to messenger RNAs, which then prevents these transcripts from being translated into proteins. So this then allows for the regulation of which genes are active in each cell, which then influences everything from cell growth to development. So maybe in simple terms, microRNAs can turn off specific genes by binding to the complementary sequence on messenger RNAs. As I said, this has kind of revolutionized what we knew before, and yeah, it's been a big revelation. And then at the end of the paper, Crick also says he's referring to, so I should say this was the original model that they suggested, but they came to realize that this was the most likely. But he hypothesizes on potential alternative theories. And in the end, he says, despite whatever the actual truth is, it would be of the greatest interest to know, but they could be accommodated into our thinking without undue strain. So whatever the outcome, whatever the truth, obviously we just accept that, and that's kind of what's happened with this discovery. So with that, I hope I've made clear how microRNAs work. So I just want to go on now to discuss their role in disease and the implications for future therapeutic development. And I'll apologize in advance for this very busy slide, but yes, just to kind of make clear that microRNAs are implicated in many different disease types, many indications, not just cancer. But so you can see cardiac disease, cardiac stress, kidney fibrosis, etc., diabetes. But they seem to have been, the disease that they've seemed to have most been implicated in is cancer. So microRNA biogenesis proteins DROSHA and dicer are downregulated in different types of cancers, and this is associated with poor patient outcomes. And of the microRNAs that are dysregulated in cancer, the MER34 family is the most researched, with MER34A, B, and C being downregulated in lung cancer, breast cancer, as well as others. And all of this family is transcriptionally regulated by the tumor suppressor protein P53 during DNA damage response. also, interestingly, increased expression and interaction of PD-1 and PD-L1 have a significant negative role in disease progression, but some studies have shown that miR-34 downregulates expression of PD-L1, meaning that it could be useful in cancer therapies. Also, loss of the LET7 family, which, if you remember, was the first evolutionarily conserved microRNA to be identified, causes cancer and activation of different oncogenes, including the KRAS oncogene, which is responsible for a very aggressive form of cancer. A final example, just in this slide, is the miR-200 family, which modulates expression of proteins that are involved in tumor metastasis and angiogenesis. So both are important hallmarks of cancer. So yeah, clearly very important. And maybe to hint at a later slide here, microRNAs are being kind of developed as potential therapies, and because they can't just be necessarily administered just as microRNAs, because RNAs are inherently unstable, they need different novel delivery systems. For example, liposomes, lipid nanoparticles, etc. But I'll talk about that in the next couple of slides. Now towards understanding how microRNAs can be developed as microRNA-based therapies. A key to reaching that goal is first understanding the biology and the relevance of the microRNA candidate to the disease. And so this can be done using tissue culture, for example, what is done with C. elegans, and in vivo model-based studies. As well, in this day and age, we have a huge amount of data that's publicly available, sequencing data. And if you dig into that, many new findings can be revealed. And also one of the major issues, as I said, for RNA-based therapeutics is degradation by nucleases and endosomal escape. And one way to improve stability is to introduce different chemical modifications, such as the three examples here. Also, several encapsulation methods, such as in liposomes or lipid nanoparticles, can be used, and this results in improved delivery, improved stability, and improved delivery to the disease sites. And lipid nanoparticles also help, improve bioavailability. Key challenges that are associated, or additional key challenges that are associated with RNA-based therapeutics is immunostimulatory effects. So potentially these microRNAs inducing immune responses or unintended immune responses, toxicity due to charge, endosomal escape, and getting to the right target. And what I'll talk about later is the use of biophysical tools and even cell-based and molecular-based assays to understand or overcome some of these challenges. But once a microRNA is identified for a specific disease and the appropriate delivery system is identified, what is needed next is rigorous preclinical testing to understand the efficacy, the toxic effects, how the microRNA affects the target, and other therapeutic abilities. And then subsequently, clinical trials are needed. As I said, because of the nature of microRNAs, they need different delivery systems to help them remain stable and get to the right tissues. So the table here on the right, sorry, I'm looking over to my big screen because it's quite small to see. The table on the right shows different mRNA-based therapeutics that are in preclinical development. Their therapeutic agent, which if they're a microRNA mimic or if they're an anti-MIR, and their delivery system. So briefly, just to talk about the different delivery systems, we have locked nucleic acids, or LNAs, which are modified RNAs, modified RNA nucleotides, which have unique structure and function. And these have the advantage of being tight binders. They have excellent specificity, low toxicity, and increased metabolic stability. Another delivery system is anti-MERS. So these are a type of antisense oligo that's designed to inhibit the function of specific microRNAs in microRNA-based therapies. So these are single-stranded oligos that are complementary to the target microRNA. So similar to this. So it would act in this way, but minus the messenger RNA. So they are single-stranded oligos that are complementary to the target microRNA sequence. And in this way, they act to suppress overexpressed or oncogenic microRNAs in different diseases, and particularly in cancer. I mentioned already LNPs. These shield microRNAs from nuclease degradation and increased stability, as I said, and they also facilitate cellular uptake because of the lipid nanoparticle. We also have GalNAc conjugation. I won't say the word because I can't say it, but this conjugation of microRNAs to GalNAc enables for specific targeting of liver hepatocytes by binding to this ASGPR protein, which is on the surface of these hepatocytes. And also just to touch on extracellular vesicles, extracellular vesicles are membrane-bound nanosized structures which are released by almost every cell type, every cell in the body into the extracellular space. And these contain microRNAs as well as other cargo, including protein, different nucleic acids, and metabolites and more. And so they themselves, extracellular vesicles, can act as biomarkers of disease. And also equally, like LNPs, like nanoparticles, like AAVs, they can be loaded with therapeutic microRNAs for targeted delivery as well. So I think that's, that's also something that's being developed as a delivery system. And now to just go on to, as I said, the biophysical tools that are available to us at Concept Life Sciences for analyzing microRNAs or microRNA-based therapeutics. But first, just to maybe, for anyone who's not familiar with what biophysics is the study of the intersection of biology and physical phenomenon in terms of the instrumentation that we have available to us. So we use different biophysical phenomenon to study interactions and physical properties. So in terms of interactions, we look at affinity and kinetics, stoichiometry, thermodynamics, and structure-activity relationships. And then in terms of physical properties, we're interested in studying size, shape, conformation, surface charge, stability, and formulation. And understanding all of these different parameters allows us to get a more holistic picture of how different therapeutic products behave, and studying these in different conditions, so different formulations, different concentrations allows us to really get a better picture of how therapeutic candidates behave. And so we're really lucky in Concept in Edinburgh, where because we have access to a variety of biophysical tools and methods to assess these parameters. As I said, this allows we're a contract research organization, as Paulina said. So having these tools at our disposal allow us to help our clients to understand how their therapeutic molecules behave. And then this knowledge allows clients to make more informed decisions about which candidates they're taking forward, which ones they're excluding, etc. So just to talk briefly about the different tools that we have, I'll start off with interaction analysis. We have 4 main tools. This is first is grating coupled interferometry. This is a chip-based method in which we typically immobilize the macromolecule. So this can be the protein or RNA, DNA on the surface by different chemistries, and then we flow over on the surface of the sensor chip. We flow over the analyte, the binding analyte. So this can be a small molecule. This can be the microRNA-based therapeutic. So in this case of microRNAs, if we if we have the microRNA in a specific delivery system, for example, an LNP, and maybe the LNP surface is functionalized to bind to a specific protein on the cell surface, we can have a recombinant version of that protein on the chip surface, and then we flow over the LNP. And this allows us to confirm that the LNP binds the target protein. And I should say that when we flow over the LNP in this example, what we have is an accumulation. We do this in a concentration dependent manner. So we have an accumulation of mass on the surface, and the software can then do its magic and calculate the affinity and the kinetics. In this example actually is as I said, on the GCI, on the wave delta instrument that we have, but instead of injecting different concentrations as you would do, as you can see here, this is on SPR, but we inject increasing concentrations of the analyte. Here on wave rapid, using wave rapid technology, what we have is in a plate or a tube, you have your analyte at one concentration, and you vary the duration of the injection on the surface. So this is what RAPID stands for. This is repeated analyte pulses of increasing duration. So quite a smart acronym from Creoptics and Malvern Analytical. Surface plasma resonance is a similar method. However, this is done on the BioCore 8K, the Cytiva instrument. The sensor chip in this case is a gold sensor chip. Compared to GCI, this instrument is more flexible because we have 8 channels, which increases throughput. And so, yeah, this is more of a high throughput instrument. And you can see here an example of an experiment. I should also say here with GCI, an advantage is this no clog microfluidics, because the fluidics are in the chip itself. So once you use the chip, you just throw it away, and you don't have it. You shouldn't have a clog in the instrument, or at least where the sensor chip was. And because of that, we can use more crude samples and more physiologically relevant medium. So like serum or plasma, which is something that we do. This just increases the biological relevance of what we're studying. And it's definitely interesting to know, because obviously when we're studying something in vitro, it's a very artificial environment in a buffer. Could be PBS. So some people can argue that PBS is quite physiologically relevant, but obviously it's not as physiologically relevant as something as if you include serum or plasma. We can also do differential scanning fluorimetry. So this is a method that relies on the phenomenon that when proteins are correctly folded in a globular form, hydrophobic residues are hidden within the core of the protein. However, when you incubate the protein in increasing temperature, the protein structure begins to unfold, and these hydrophobic residues become exposed to the solvent surface. So when you have this system in the presence of a dye called CYPRA-orange, there are other ones as well, but CYPRA-orange is the most common one. And this dye has affinity for hydrophobic residues. And when it binds hydrophobic residues, you have an increase in the fluorescence. And once you have completed an experiment, you get this this thermogram, which shows the unfolding of the protein along temperature gradient. And the midpoint of this curve is called the TM. So that's the point at which 50% of the protein is unfolded. So we can use this to assess the effect of different formulations and different storage conditions. And in the case of microRNA-based therapeutics, this is useful to know if you want to look at different LNP formulations, different LNP formulations, different buffer formulations. If you want to see how different sequences of microRNAs affect stability. And then the gold standard for biomolecular interactions is isothermal titration calorimetry. And I say it's the gold standard because in this method, the interacting partners are in solution, whereas with GCI or SPR, one interacting partner is tethered on the surface. And here with DSF, we're relying on a fluorescent dye. So those features of these assays can influence binding. So these assays come with their own caveats, whereas ITC is just, you know, your two or your however many interacting partners you have in solution. And you're measuring, what you're measuring here is you have your sample in your sample cell, and we have the reference cell, which usually contains water. And so we're measuring the temperature that's required to maintain the sample cell at the same temperature as the reference cell. And this is based on the phenomenon that when there is binding, when there's binding of two interacting partners, there's a release in energy. The only one negative with ITC is that it requires quite a high amount of protein or even RNA. And if you know anything about producing recombinant protein, you know it's often difficult and or expensive. So often people use it as you know, the last or one of the last assays that they use to confirm the affinities of their products, of their candidates, sorry. And to look at biophysical characterization, we can use dynamic light scattering and electrophoretic light scattering. So as the name suggests, both methods are based on light scattering and Brownian motion. Well, DLS is based on Brownian motion. And so we use these methods to look at particle size and surface charge, and this allows us to understand stability, solubility, and interactions as well. DLS is non-destructive. Measurements are very fast and they have low sample consumption. Another method, really useful method that we have is size exclusion chromatography with multi-angle light scattering, SECMALS. So this is a chromatographic technique in which we separate samples or we separate molecules within a sample in a column. So this allows us to get information on the molecular weight, molecular size, aggregation, and purity. And when it comes to extracellular vesicles, for example, this is a really useful method because usually when you have your sample or your starting medium, for example, which contains your EVs, you want to separate them out, and often you'll have different sizes of EVs, and sometimes different sized EVs will have different, or often they will have different content, and therefore they will have different effects, functional effects in cells. So you want to thoroughly or accurately separate those and then analyze those effects. We can also use differential scanning calorimetry, which is another calorimetric technique similar to ITC, but this time we're measuring thermal stability, and this is similar to DSF, but there's no dye involved. It's just based on calorimetry. Yeah, this is a gold standard for biosimilars and biosimilarity and biocompatibility analyses. And finally, we can look at nanoparticle tracking analysis. And this is another way to measure particle size and distribution of various sample types. So this is high resolution, more higher resolution than DLS. It's non-destructive, and we can obtain dynamic information about particle movement and behavior. And one of the things we're really keen to do within the biophysical sciences team at Concept is to use involve integrative analysis during in different projects, because we've seen the benefit of analyzing one sample with multiple different techniques, because that can reveal a lot about how the sample is behaving, and even more if you compare one candidate to the rest. So one candidate can be an antibody, or a set of candidates can be, you know, a set of antibodies that are slightly have different mutations, or different microRNA based therapies, maybe with different LNP formulations or different sequences. And looking at the big picture of how these molecules behave, these candidates behave in comparison to each other, allows for to mitigate late stage failure, and then allows for more informed decision making. So, because if you can look at affinity and say, yeah, this is great, this is, you know, it's a tight binder, but when you look, for example, at, you know, size distribution and conformation, the size maybe of the LNP varies because it's aggregating or it's in a weird conformation, and you wouldn't necessarily know that if you didn't look at the different methods. So yeah, looking at multiple different parameters saves time and helps with downstream prioritization. So with that, I hope I've made clear the utility of biophysical characterization in general, and also in terms of microRNA-based therapeutics. One of the things we have developed is our INSERT platform. This is the platform that the INSERT stands for in vitro characterization and efficient RNA transfection. And this is a customizable workflow that we've developed to allow for our clients to interrogate their cell and gene therapy products to analyze them using biophysical, molecular, and cell-based methods to look at how these different parameters that are analyzed here, how they affect function. Again, to basically allow clients to make informed decisions for how they move their candidates forward. This is my second last slide, just to kind of show you, just to emphasize the fact that we are an integrated company. We have different service lines. I didn't say before, but in Edinburgh, in biology, we have 4 different service lines. We have biophysics, neuroscience, spatial biology, and immunology. So we're kind of really well placed to analyze, to help our clients to kind of look at many different aspects of their project. We also have a chemistry team and in England, in Chapel and Sandwich, and we also have a toxicology and bioanalysis team in Dundee. And so using all of our the tools at our disposal, we're really excited to help our clients advance their products from concept through to clinic. So with that, I'd just like to thank Sano for the opportunity to present here. I'd like to thank Carolina and Paulina for facilitating, and my colleagues at Concept. And as I said, I'm not a microRNA expert by any means. I hope I've been able to clarify or convey how this discovery came about, its importance in general in biology, and how it can be exploited in the future to advanced medicine. Feel free to contact me at the email there if you have any questions, if you'd like to know more, if you want the slides, I'm happy to provide them. Yeah, thank you for your time.
Thank you very much for your talk. And now we have time for question, for discussion. Well, maybe we have already Adam's work, but I would like to ask the question about a relation between Nobel Prize in 2024 and Nobel Prize in 2023. It seems that it is a kind of a chain, because it was a Nobel Prize which was given in 2023 was something about nucleoside modification, and here is also about modification. So, well, what is your opinion? There are so many other, let's say, places in RNA to get next Nobel Prizes.
Yeah, I think it's maybe just because I believe the Nobel Prize you're talking about is the one for LNPs, right? Like mRNA based vaccines, right?
Yes.
Yeah. I guess it's maybe just following on from how important that discovery was. I think these discoveries, like I think even for that prize, this knowledge had been around for decades, but it only came really into popularity, or really came into the public consciousness when COVID came. So maybe it's kind of what's popular at the time, but I guess, or I assume, that the deserving prizes will still happen at the right time. It's just maybe, yeah, microRNAs or RNAs are hot at the moment.
Okay, thank you. And now we have Adam's work. Adam, please go on with your question, comments.
Yeah, so my point of view as a chemist, Well, when we are trying to develop new chemical small molecules, which could be possibly anti-cancer drug, there is a big problem with off targets, because many of one thing is delivering of this molecule particularly to cancer cells, but another thing is we are hope that mechanism of for example, some proteins are more abundant in cancer cells, so that's why we trying to act on this particular molecular target. But many times the problem is with selectivity, because this drug can reach to every cells and could causing damages. What's about this method? Because you mentioned that silencing of some oncogenes could be beneficial, of course, but what about harmful effect of this?
Yeah, so like unintended effects. Yeah. To be honest, I've not really read into that part yet, but I think, yeah, with every therapy, there's the risk of unintended side effects. But I think, for example, with the LNAs, the locked nucleotides specifically, they're the affinity that they have for their target microRNA is based on that, yeah, based on that specific sequence. So I think they're trying to reduce any non-specific effects, but I don't think it's necessarily possible to get rid of them completely. But maybe compared to small molecules, they may have fewer unintended side effects. But I have to confess, I've not read into that in detail.
But do you have information on which clinical stages they are already?
For the things the ones that I discussed?
Yeah, like do you have any example, like is this first clinical trials, or maybe they are in the market in the clinic?
I don't think anything is really in the clinic yet. I think most things are still undergoing clinical trials for microRNA.
Yeah,
Yeah.
you mentioned this.
Mm-hmm.
And maybe another question, more general, what is your intuition about the future therapeutic goal? It will be immunology, or maybe this method, or maybe small molecules?
Well, tough question, Adam. Tough question. I think novel modalities in general are well, yeah, I think in recent years, novel modalities have kind of been the most popular. So PROTACs, for example, LNPs, like I said, AAVs. So I think because of the disadvantages of the small molecules, their non-specific effects, the screening cascade, the attrition rates, maybe that's kind of led, and also the advances in biology, considering something like this, is maybe why we've seen this increase in the novel modalities. But I think there's still a place for small molecules. Yeah, don't know if that answers your question.
Yeah, thanks. Because many times I heard this strong opinion against small molecules, but so far we see that they are in use, and probably they will be for a long time with us.
Yeah, I think there's definitely still a space for small molecules, despite what people like, all their disadvantages. I think there's still a space for them.
Yeah, thank you very much.
No worries. Thank you for your question.
And they are in the title of Zan talk, they are titans, so maybe they are small, but quite
Small but
large.
mighty. Yeah.
Yes. Olaf, go on, please.
Yeah, thanks for the talk. So, well, when I was working with RNA, so my main concern was it was so damn unstable. Yeah, getting a northern blot really on the gel was always you had to be very lucky if this not was half chewed up by RNases. And in your experience with all your physical techniques do you have, so how do you see handling of RNA? Okay, I saw in the therapeutics you were introducing that they have modified them to make them more stable. Yeah, so because that is also, of course, an issue. You want them to act before they are chewed up. And so how do you handle these complex types? So I anticipate that also the mRNA, the microRNA therapeutics, they somehow have to bind this risk complex, so with ARGO2 and the other proteins that are typically involved in microRNA activity. So how would you measure such affinities or so in your lab? So you can't do this with microRNAs alone. You would have to do the entire complex. Is that something that is an established technique already?
We haven't done it ourselves, like looking at ARGO2 and microRNAs, but it's possible, right? So you can break apart the different components and look at maybe just the RNA and the microRNA and the RNA, look at the affinity of that, and then bring in another interacting partner and build the assay that way, and look at the affinities of different things together, and that can give you a bigger picture of what's happening. In terms of how we handle them to kind of prevent aggregation or degradation, sorry. Yeah, we try and be as careful as possible, use the standard methods, kind of have careful assay development. So do it, one batch at a time, because some of our assays can take a long time. For example, the DSC, depending on your assay parameters, you can have multiple hours to run one sample. So obviously we wouldn't want to set up 10 RNAs at once and leave them to go, because that could take a week to run. So it's just careful assay planning and trying to do things in the most logical way as possible in an RNase-free environment.
And can you do this? Like in this case, a protein RNA complex that together has an affinity for the target protein or target messenger RNA. Can you measure that while keeping the complex more or less assembled? Or these biophysical techniques very often are, well, not gentle to molecules, to say the least. And
Yeah.
sometimes, especially these kind of complex things where a complex is the acting agent and binds to something, I imagine it's totally difficult to measure anything without disrupting the interesting interaction first. So
Yeah, you're right. Yeah, the interaction itself has to be very tight, right? Because as you say, we're using sometimes high flow rates, high stir rates, sometimes high temperatures, but it is possible depending on the system. If you have a really nice system, it's tight binders, and you look at binary complex, for example, and then bring in ternary complex, it is possible to do, provided that the affinities work. So
okay.
there's always a
Then what would
caveat.
be what would be your favorite technique for doing that from the arsenal you were presenting?
Probably SPR, for
Okay.
example. So you can maybe have, depending, you can do different assay setups. You can have like the protein immobilized, then flow over the RNA, and that if it binds and that's stable enough, you can flow over the third partner, and you'll see that change in surface accumulation. But you can also look at binary and ternary complex formation by ITC. But as I said, that requires a lot more protein, a lot more reagent. Yeah.
Okay. Thanks a lot.
No worries. Thank you.
Thank you very much. And well, there was in discussion with Olaf, you touched the problem of, let's say, research method. So what is the place of computer simulation in the research method? Do you, is there something which can be useful for in this area?
In biophysics, yeah?
Yes. Oh,
Yeah.
yes, especially with these small molecules, yes.
Yeah, so I imagine there's something like alpha. I don't know if AlphaFold works for RNA sequences, but something like that to maybe understand maybe how mRNA folds and how the microRNAs can bind is useful. But as with anything, models are great, but you only prove it once you do the experiment, right? So your model can tell you one thing, but when you do the actual experiment, it can be completely different. So I think it's useful to have if it's available, so that it's always nice to have additional context when you're planning an experiment, so that you can plan it as thoroughly as possible. But yeah.
Yeah, I can add to that. So AlphaFold 3 can do RNA, but the database is only a fraction of what we have in experimental data that we have in proteins. So that is with some kind of grain of salt in it, and all RNA force fields are crap.
Well, don't be so cruel.
There's your answer, Marion.
Okay. Well, on your screen we see this diagram of let's say all these processes which lead from concept to clinic. Well, could you please put let's say time axis on this? So what if we start on, let's say, 1st January 2025 with concept, how long we should wait until, let's say, we successfully end in clinic?
I won't get myself into trouble by saying a number, Marion, but I believe I think we had a statistic on LinkedIn, but related to how our chemists help our clients get their product to clinics. Obviously, our chemists often either do the large-scale production of a small molecule, for example, or yeah, once they get to the large-scale production, and then that goes into the clinic. So I think with that one, that's maybe a more realistic number. I can find it. I think it was maybe 10 years. I can't remember, but I will find it and share it with you. But in biology, nothing is ever simple in biology, so I wouldn't put a number on it.
Yes, especially with Olaf's comments.
Yeah.
Thank you. Well, I don't see more questions. So many thanks, Zan, for your talk. Many thanks for taking part in discussion. Thanks to Olaf and Adam. And I hope that we will soon see your talk also on in recorded version, and for colleagues who had no chance to participate in this. So many thanks, and kind invitation to Krakow all the time is valid. Thank you.
I will be there. No, thank you again for this opportunity, Paulina, Marion. Hope I didn't say anything completely wrong. I don't know, but
no, it was great. So thank you very much for all participants in of this seminar. It was seminar number 150 from the beginning of, let's say, of Sano seminars. Well, this year we celebrate something like 5 years of operation of Sano, and and it was last seminar before Christmas. The next one will be on January 13. It will be a kind of a Christmas and New Year break. So I would like to wish you all the best for that time, and also for the new year. And I kindly invite you to next seminars of Sano. Thank you very much, and see you soon.
thank you.
Thank you.
Thanks, bye.
Bye.